once infectious elemental bodies infect the target cell, they transform into reticulate body to be able to replicate. And the reticulate body we replicate by binary fission, like other bacteria, and the replicated, again replicating, it will fill the cell. And by filling this cell, there will be transformation from reticulate body to elementary bodies. Because gender the elemental bodies, then they will be excreted or released from the cell to the outside environment to infect new cell. They have hemagglutinin that allow them attachment to the cell, and by cell-mediated immune response, they cause damage. So the damage is more of reaction of the immune system rather than the pathogen-related virulence factors. So although an endotoxin like toxin is involved, endotoxin, as this is gram-negative, ariko mostly cell-mediated immune response niyo igira damage kubera ko izi infected cells zose displaying the antigen outside, then the immune system we want to clear everything over there. So trachoma occur worldwide and is prevalent in Africa and Asia as most infectious disease, and it is usually inoculated in eye by contaminated finger or fomite. So mostly in neonates, but passed through an infected birth canal, mostly young anaboka. So genital infections, they were spread venereal, donc by sexual intercourse, and respiratory by inhalation, droplets. And psittacosis or psittacosis is acquired from infected bats. Diagnosis, as we have seen, it doesn't grow on acellular media. So the most reliable is immunological and PCR, nucleic acid amplification. Sometimes for studies you can do tissue culture technique, and you can also be able to visualize the organism in epithelial cells by GEMSA or iodine stain. The smear, you can see how the parasites have really grown in a single cell. Treatment is easy. Tetracycline and erythromycin are the drug of choice for this. So besides chlamydia, the mycoplasma, and these are the bacteria without cell wall. So they don't have the cell wall by nature. And among these, we have two important genera, species. One is Mycoplasma pneumoniae, which cause upper and lower respiratory tract infection, inducing cough, fever, headache. And you have Ureaplasma ureaticum, which cause more non-gonococcal urethritis in male and urethral discharge associated with this, while female remain asymptomatic generally with this. So hii pathogen na Chlamydia trachomatis, they are the causative agents of non-gonococcal urethritis in men. So umuntu ababajije ati what are the most or the major cause of non-gonococcal urethritis in men, just remember ureaplasma, ureaheticum, and Chlamydia trachomatis. They don't have cell wall, as we said, and they have a tip of filamentous organelle that allow them sticking on the cell. So this is the organism. The genetic material are quite cold in this sense. You have lipoprotein of the membrane and many soluble protein. And it's just simple as we can see, but potentially causing disease, either lower or upper respiratory for Mycoplasma, pneumonia, Mycoplasma genitalium, that cause more of non-gonococcal urethritis. So they have the smallest genome, and they are of course self-replicating organism. They only have this very low number of gene, and they have low GC content. They are nutritionally very exacting, so that means they are very demanding. They are very fastidious, and they may require some special component nutrient like cholesterol and urea, which is rare for bacteria, normal bacteria. They have surface antigen. Some of the membrane undergo antigenic variation. So I think we have seen something like this somewhere else. Who can tell us? Antigenic variation, which other bacteria that we have seen having this property, expressing this no? Which other bacteria that you have seen to be able to undergo antigenic variation? And what is the role or importance of antigenic variation? No? So the group that presented is entirely absent. Badu high presentation harimo antigenic variation. Nta n'umwe uhari? No? Hello, can you hear me, please? Gahire, can you hear me?
Yes.
So can you tell us?
I think the other bacteria is Neisseria gonorrhea.
Neisseria gonorrhea does not have this property. Who else? Brigitte? Irea? Brigitte, are you there? No? We have seen that kombona tuzagirana ibibazo ra. I have seen that some of spirochetes have this antigenic variation, and one of the typical is the causative agent of relapsing fever. Sibyo se? Relapsing fever ntago muyibuka? With Borrelia recurrentis. So I think you will be able to connect. So this is a very major property that really evades the immune response. The immune response imaze kuba established, the pathogen undergo the antigenic variation, then the immune response we miss this. And the host we initiate while the organism. Then once the immune system is established for the new antigen, then the pathogen change again. This will limit the ability of the organism to really eliminate the infection. So Mycoplasma, the parasite human respiratory and urogenital tract, and they attach to cycloglycoprotein receptor, and they produce free radicals, like hydrogen peroxide, A2 the cause of non-gonococcal gonorrhea. So by having this as a reservoir, then the spread is continuously within this circuit, as sexual intercourse ensure the transmission continuously. Hiyo ni more of the colder area, the Mycoplasma pneumoniae, and is more prevalent in colder months, in winter and spring. So as diagnosis, you can use sputum, and the base is more serology test that you can use, or you can also do DNA or PCR to detect the pathogen. Treatment with tetracycline or erythromycin is effective in reducing symptoms. There is no vaccine, so it's more antibiotic that can cure the patient. So before we do mycobacteria, I would briefly break, have a break of 10 minutes. So we come back at 10:40. Tugaruke saa 4 n'iminota 10 for the next session, where we discuss more on mycobacteria and other bacteria of medical importance. So let's break for 10 minutes and come back. Question after break. Are you back or can we resume? Hello? Murahari tusubukure? Hello, hello. Murahari mwagarutse? Are you back?
Yes.
So Mycobacteria. Mycobacteria is an interesting class of bacteria which is more special on its own, having a very unique cell wall. And the cell wall of Mycobacterium offer a very specific features that we see it make even difficult manage TB. So the cell wall of Mycobacteria is more of gram-positive first. It have peptidoglycan layer, but beyond peptidoglycan layer, there is a big layer of mycolic acid, free lipid, glycolipid. So this layer of mycolic acid, which is a strong component of Mycobacteria cell wall, we limit many of the antibiotics that we can use. Also making this pathogen to be more complex to treat. So we have three important genera. We have tuberculosis, the Mycobacteria tuberculosis, which is the causative agent of TB. We have Bururi Ilsa, the causative agent, or we see Mycobacteria ulceras as and we have leprosy as a disease resulting from Mycobacteria leprae. So the three important species are the main pathogen, although others can also be opportunistic pathogen. It have gram-positive like cell wall structure, it have peptidoglycan. But what is special to this, it have 6% of lipid in the cell wall. It is not spore forming, it is not capsulated, and it is strict aerobe. So main lipid component in mycolic acid, NACOD factor, Changa wax D. So these three important content of lipids confer some property that we are going to see. They don't produce exotoxin, Changwa endotoxin, and the disease they cause is more induced by the immune system reaction. So, and is more of delayed type hypersensitivity reaction to mycobacterial protein. Delayed type hypersensitivity reaction, immunology, it's really a immune system reaction. Yugayo itera ikibazo. It's hyper in high stimulation, so it causes destruction of the area. Ibyo akaba ari byo bitera ikibazo kuruta uko bacteria yari ibereye aho ngaho idafite toxin ariko ikenducinga iyi hypersensitivity, high reaction of immune system. So it elicits a granulomatous response, causing chronic course of disease. And we see, as we said, high lipid content confer impermeability to stain and dye to the cell wall of microbacteria. It causes resistance to many antibiotics. Many antibiotics cannot get in the cell, and resistant to killing by acid and alkaline compounds. So it can't be killed by acid or alkaline, kubera ko ziriya layers za lipid zibigarura, and resistant to osmotic lysis via complement the composition, resistant to lethal oxidation and the survival inside of macrophage. This is more, these two are more beyond the cell wall. Mahano usanga ari the property iri within the cell. Classification dufite class ebyiri, hari obligate parasite cyangwa depending on isolation, hari iyo bita immune grouping that can be used, but for now it's more old classification, this one. So we have M. leprae, which is not or non-cultivable, and we have M. tuberculosis complex, IRIMO, some of the species, and M. ulcerans, which grow based at certain degrees. Izi eshatu nizo major pathogen za microbacteria. Izindi tuza kubonamo hano, they can cause disease, but they are not really the real pathogen. So they cause disease depending on the host immunity. So we have this grouping. You can read through. There is photochromogens that produce yellow-orange pigment upon exposure of colony to light. We have scotochromogen. They can produce pigment either in light or dark. We have non-pigmented, and we have rapid growers that grow rapidly in the lab. Microbacteria grow very slowly in media, egg-containing media, so they can be differentiated. So one of the important remark on TB is that it has caused as many as disease as or even combining all the pathogen. Hagozagera aho TB igeze yica abantu. Sibyo, mu myaka magana abiri ishize, around 1 billion people worldwide have died from TB. This is really a big number. And as I said, a complex microbiome tuberculosis, the causative agents have all these species in the complex, and it's all this disease, and can affect lung mostly, but other organs are also involved. So you can see how comparable the disease, even HIV, ahigeze. So you can see how much TB is really a serious disease. Infectious disease. Kuko iyo tureba izindi ndwara zica abantu cyane, akenshi usanga ari NCDs, non-communicable disease, nka cardiovascular, ari nayo ya mbere. So it's muri infectious disease iri muri number 1 killer. Mode of transmission ni person to person, inhalation, pulmonary TB, sometimes ingestion of milk, kurugero, infected cow, iyo zigize zishobora ku-infecting umuntu bacteria, or sometimes by touching. If it abrasion, then when you manipulate the bacilli, for example, in the laboratory, you can have the skin infected with microbacteria, zikagrowing zikatera ikibazo. Risk factor, iya mbere ni immunosuppression due to different causes, including HIV/AIDS, malnutrition, some behavior like smoking, alcohol, diabetes, and all these. Gender, male are more susceptible than some overcrowding, of course, poor poverty, which is more criteria. Majority of these linked to poverty. Of course, you know all that the overcrowding, the malnutrition, alcoholism, smoking. So the disease used to be like understanding as a disease or not disease, but now the spectrum seems to be a bit large. So iyo umuntu abaye infected na TB, there is a very short incubation period. Sometimes it can be short to have pulmonary TB cyangwa primary TB, ikaba ihita developinga mu gihe gito umuntu aba infected. Sometimes it remain as latent. Latent means you don't have symptoms, but the bacilli are within the lungs, they are live, and sometimes it can immediately be eliminated. So these are 2-3 possibilities. It can be eliminated, infection eliminated. It can be latent, or it can just develop as disease, symptom. So this is the R phase. So sometimes the latent TB may be activated to produce symptom. That is what is called reactivation. And sometimes the reactivation is vuba, which is the most likely scenario. Iyo itinze cyane kenshi ntabwo igira. So some others have shown that TB can be a cyclic disease where some few symptoms can appear and disappear and come back and go continuously like this. So this is a theory that is being explored by different researchers, whether this is real. Then we have the slow reactivation, which can come years later after infection, and it can also be eliminated some years later, or the person can remain infected for life. So we have a case of TB eliminated, that is the best case scenario. We have latent TB, which is a continuum infection throughout. We have incipient TB, that is where you have that kind of cyclic disease. We have subclinical state. Subclinical state, you don't have such clear symptom, but you really close to active TB. So all this is the spectrum that is more being investigated for now as far as TB pathogenesis. Clinical presentation, mu Rwanda tugira 8% in pulmonary, some others tugira extrapulmonary in less than 20%. And we have cough as a symptom, chest pain, hemoptysis, fever, night sweat. These are really the typical characteristic of TB. And extrapulmonary is also turazibona. So with diagnosis, we use microscopy, but nowadays we have real-time PCR, automated real-time PCR machine tuza kureba mu kanya. So as microbiological investigation, first, mbere yuko bagira na microbiological investigation, clinician agomba kuba agomba kuba yagize neza differential diagnosis, ashobora kugira iyo suspicion ya TB. Dufite ikibazo cy'uko rimwe na rimwe abaclinician badasuspectinga TB muri symptoms umurwayi aba yababwiye. So that is a critical condition. So then the sputum have to be appropriate, the sample have to be appropriate bitewe n'uko umurwayi arwaye. So lymph node biopsy cyangwa aspirate, fine needle aspirate, body fluid. So sputum is the common. So the sample should be really taken from the area that is linked to the symptom of the clinical manifestation. And the microscopy is more used to dukoresha oramine nkuko twabivuze, ya servo ya mycobacteria confer impermeability to stain, and that is why the background is seen as different from the bacilli. Even here we have the bacilli in red and the background in blue. This is because these are acid fast. They resist to the colorizing by acid due to high content of lipid in the cell wall. Same here, they retain oramine, and oramine is a fluorescent dye that reflects light. So niyo mpamvu tuzibona zifitiye features. So you can do culture. We have a solid culture system, we have a liquid culture system. Liquids are a bit faster than solid, and solids are more ahatureva is in colonies, billion of bacteria form colonies. By dividing, uko zigenda zidividinga ziguma hamwe zikagira mass visible by naked eyes, and this is made of millions of bacteria. So we have another system, dufiteho more gihugu gene expert, which is more an automated system igira detection ya DNA ya bacteria ya TB muri sample. Hanyuma ntigarukire kuri DNA ya bacteria gusa, ahubwo ikavuga niba harimo na mutation, harimo likelihood yo kuba iyo bacteria iri resistant cyangwa iri susceptible. So it's important to know that iyi twayigizeho amastudy menshi. So this is my Google Scholar. If you click, you can be able to select what you want to read, as all my work, my publications are in here. You can read much more on experts. Twagizeho ama analysis menshi to demonstrating some of false results. Tunakoraho documentation on how this technology have really improved the management of patients with DRTB or rifampicin-resistant TB. So you can read on some of our article and get to know what is going on as a program concern, TB program concern in Rwanda. So dushobora no gukora MANT test. We can do tuberculin test, skin test. So before I forgot, maybe it is also important to highlight that in our work we showed a very special strain So among others, mwaza no kubonamo iyi article, ni article yacu twadiscoverinze a new TB strain to documenting. So it's really important to know that we showed this strain to be more basal. So our strain was here. We showed this strain to be more basal and documented kind of the origin of suggesting the origin of TB mycobacteria in our region, in our East African area, where we also think that humanity came from. So the TB went from our region with migration of. So you can read the article. It's our discovery. You can read. It's published in Nature Communication. You can have more information on this if you wish. So otherwise, you can also read on more za gene expert twazikozeho cyane, twerekana ibyiza zagiye zikora ni na challenges tugenda tugira. So twerekanye ko iyo ukoze diagnosis kare ugira interruption ya transmission ya TB. So unfortunately, many of TB patients are not diagnosed. In 2019, only 30% people remain, cyangwa up to 30% y'abantu ntabwo babaye diagnosed. So no kuba diagnosed 30% ntibaba diagnosed kandi barwaye. So they keep transmitting the infection. So again, only abantu bari munsi ya 40% nibo OMS ivuga ko bagiye ku miti muri 2019 iri appropriate ya resistant. So meaning that the rest are more than 50% are still there and transmitting. So we do have two level of treatment. Hari first line, duha abarwayi bose. Hari na second line. So we do second line for patients with resistant first line, principally rifampicin. Your rifampicin resistant, this first line will likely be unsuccessful. Then we need to second line. How to correct more drug and more longer periods. So you can read the definition to multi-drug resistant TB in case of resistant to rifampicin in isoniazid, which are the major or the main component of the first line regimen, and extensively drug resistant TB in case TB is resistant to these two, plus fluoroquinolone or bedaquiline and or linezolid. In this case, then the patient will be unlikely or likely untreatable. So the treatment of resistant TB should be based on susceptibility results. So the laboratory is important to really show what drugs are still effective to treat the patient. Control the correction BCG, based the candidate guerre, although it's no longer effective as, and it doesn't really protect at late age. To only protect the child, newborn, from the serious, severe condition like meningitis. Ariko ntagikomeza kubarinda throughout. So you can do isolation, you can treat case, you can do isolation of patients with ventilated household, contact tracing kugira ngo ugabanye infection, sometimes chemoprophylaxis, IPT, turayikoresha cyane ku bantu bari more likely to developing a TB. Any question with TB? Or other aho twahere hari ikibazo cyihariye mwaba mufite? If not, we continue with Microbiological Lecture. Twabonye ko ari more obligate intracell organism. It cannot grow on acea culture media, only in the mouse parts or in the armadillo. So it have to grow on living cell. Why we see why is they are not able to synthesize ATP, they cannot synthesize energy. So reservoir infected human, and they have a very low infectivity, so they don't transmit as high as it should. Skin to skin contact also is frequent, but respiratory route is also possible. Niwe wari Musanze? Niwe wari Musanze? Niwe wari i Rubavu? Ndakwandikira urahita uyibona, sibyo? Sorry, and skin-to-skin contact, but low infectivity. So whenever you touch a patient with leprosy, don't expect that you are going to get the infection. And sometimes skin, of course, nasal mucosa is the exit. And the incubation can be up to 30 years. So it can have that very long incubation period. And it have tendency to affect the cooler part of the body. So why is that? You need to find out why does this really prefer the cooler part of the body. So we have 3 or 4, 5 clinical stage. One is, or the initial is where the cell-mediated immunity is the highest. Tuberculoid leprosy, hakaza borderline tuberculoid, hakaza mid borderline tuberculoid, where there is quite equilibrium between cell-mediated immunity na antibody response. Ukuvuga iyo gitangira ni more cell-mediated, uko igenda imara igihe nibwo antibody responses is borderline lepromatous and lepromatous leprosy, which is the latest stage. Iyo urebye kuri clinical pictures cyangwa description hano nibwo ubona differences. So this is tuberculoid leprosy, where you still have these patches of lepri, which is not really characteristic. Then lepromatous leprosy, utangiye kugira some of advanced lesions zigaragara cyane. And bacillus density is heavy. Lepромine reaction, this is the antibody reaction, is known. So ahani more cellular mediated immunity. We have classification by WHO. You can read all these. Diagnosis is more clinical. There is a clinical criteria. You fit 2 of the 3, then they can clinically diagnose leprosy. One is anesthesia of the skin, so the skin gets an anesthetic. So thickened peripheral nerves or typical skin lesion. Any 2 of these 3, then you have the clinical diagnosis. You can also do a smear. Ukaza kureba the occurrence ya leprosy ku bantu babana n'abarwayi bafite leprosy. So Mycobacterium in itself, the causative agent of Virulence is also more pathogen in Mycobacteria. And Virulence is related to area that flood with swamp formation. So haba swamp cyane niho usanga Virulence. One is you have 4 levels. There is a subcutaneous nodule, vasculitis, and spreading cellulitis, then ulceration, healing with cross fibrosis. So this feature iba yuzuyemo za mycobacteria. So igikora izi lesion ntabwo ari toxin ziba kuri bacteria, ahubwo ni immune response reaction kuri izo bacilli zabaye ziri kugrowinga muri environment. So clinical features can already show be used to diagnose the patient. Acid-fast bacilli in smear from basis of ulcer, biopsy, culture, PCR can also be done. And treatment is chemotherapy is not effective. So you need really to do excision. Sometimes you can do more or very wide excision, requiring even grafting, skin grafting. Kubera ko niba umuntu yatakaje big part of the skin, then it will require some grafting to ensure the reparation is done. Any question with Mycobacteria? Any question? Turikumera, can you hear me?
Yes. Yes.
Okay, spirochetes. So spirochetes ngira ngo nazo twarabivuze ko hari group yari presentinzeho very well. So we have three important genera. We have Treponema, hari nayo irimo the causative agent of syphilis. We have Borrelia, where we have Borrelia lecrantis, the causative agent of relapsing fever, and we have Leptospira, the causative agent of leptospirosis. I think these have been discussed, but we have more visual of. So spirochetes come from Greek, like coiled hair, spirochetes. Thin and can be very long, so they are very thin. We have three important genera, genus nk'uko twabivuze, Treponema, Leptospira, and Borrelia. In three families, cyangwa two families, I don't know. Dufite Spirochetaceae and Leptospiraceae, where Spirochetaceae dufitemo Treponema na Borrelia. So we will not do this too much. Coiled, murayibona hano uko iri coiled, the fibril, and this is the end of flagella, as we said. And here turazibona hano, so there are many, and they can see within the periplasmic membrane. They are enveloped in sheath. So that is how the pathogen, we just use this flagella to move the entire body outside flagella. So we have some species and subspecies, Duphyte Treponema, which have around two species, Pallidum and Carateum. Pallidum, the causative agent of syphilis, and Pallidum endemicum or Bejer, which is also called endemic syphilis, and Pallidum perteni, which is the causative agent of VIAS, and the Pinta, which is caused by Carateum. I think we have seen all these. Borrelia, Duphyte bulgotropherin, which is the causative agent of borreliosis, cyangwa limb disease. We have Reclantis, Borrelia reclantis, the causative agent of epidemic relapsing fever, and other many species which cause endemic relapsing fever. Epidemic relapsing fever tuzabonako iri linked na vector, which is more the disseminating factor of the endemic, and we have the Leptospira interrogans, the causative agent of leptospirosis. Let's start with the non-venereal treponema disease. Ya mbere ni endemic syphilis, caused na Treponema pallidum endemicum, Bejer also. Initial region, non-descriptor oral lesion, mostly oral, and there are patchy skin, bone, and nasopharynx, especially at late stage, and it's transmitted person to person through contaminating utensils, eating utensils, ibiyiko, frusheti, spoons, forks, and all the eating utensils can be the cause of transmitting this. Question, Muhir? You can't see the slide? Sorry. You still don't see the slide? Now you can see the slide. So it is transmitted person to person by contaminated eating utensils, and it is more prevalent in primitive tropical cyangwa subtropical area in Africa, cyangwa Asia, Australia. So it is common in the yours, which is more granulomatous disease. If you can see how it really causes some granulomatous reaction. Skin lesion in late stage, they are destructive lesion of the skin, lymph nodes, and bones, transmitted by direct contact with a lesion containing abundant spirochete in here. And they are mostly in South America, Central Africa, Southeast Asia. Ini is pinta, which is caused by another species, Carateum, and also small prelytic papule lesions leading to plaque for months or years. Also hypopigmentation in late stage, the pigmentation of skin, disfiguring, the color, the entire color, and they are transmitted direct contact, skin lesion, and they are more in Mexico, Central South America. So beside this, which are non-venereal treponema disease, then the venereal or sexual transmitted treponema disease, which is caused by Treponema pallidum, subspecies pallidum. So as you can see here, this is a stain of the spirochetes. You can see in dark field microscope how they look like, and it is primarily transmitted sexually, sometimes transmitted through or vertical during to the fetus. There are two scenes. Sustain. They are very seen. Can be seen with a dark field microscope, staining with anti-treponema antibody label with fluorescent dye. So when you stain with antibody, because getting an fluorescent dye, they will of course reflect light, and nazo tulikuona zimera nk'iziri kwaka. So they are intracellular pathogen, cannot be in cell-free culture. So that means they really they don't certify coaches nk'uko twabivuze muri ya presentation previous. They don't survive outside the host cell. So that is why if you really need to do laboratory culture, so you really have to take care of specimen to make sure they are still viable. They are transmitted sexual contact, cyangwa mother to fetus, and they are not highly contagious. Only 30% of chance from single exposure ugiranye sexual intercourse na infected partner. But transmission rate depends upon stage of disease. Long incubation period, niyo iri more characteristic, during which time host is non-infectious. During this period, incubation infection ibaho, donc is non-infectious, and useful epidemiological for contact tracing and administration of preventive therapy can then really cut off so niho prostitutes sibyo yakubwiye network so you can easily treat and cure or break the chain of transmission. Virulence factor ya mbere ni outer membrane protein promotes adherence. They have hyaluronidase enzyme. They have antiphagocytic coating. And the tissue destruction and lesion are primary of host immune response. So the immunopathology, typical, similar, or quite close to what you have seen with TB. Destruction ya skin, ya area, changa, ya symptoms, we result more from immunopathology. That means immune system or immune reaction will be the more origin of disease. So pathogenesis of T. pallidum, tissue destruction nk'uko twabivuze is more a consequence of patient immune response, and they will be more extensive in blood vessels. Syphilis is a disease of blood vessels in perivascular area. So to destroy the perivascular area and blood vessels. And in spite, the host immune response are capable of persisting for decades. Nubwo immune response very high cyane kubera ko izi pathogens zijya muri cell intracellular, so they can evade the immune system. In such, they can stay with the host for decades. They can stay with the patient for decades. Infection is neither fully controlled nor eradicated. In early stage, there is an inhibition of cell-mediated immunity, inhibition of abate in late stage, hence the lesion tend to be localized. So at the start, ntabwo cell-mediated immunity really pronounced cyane, rather the late stage nibwo tugira pronounced cell-mediated immunity. So as primary syphilis, it starts with chanco and usually painless. So it involves mucous membrane, rapid multiplication, wide dissemination through perivascular, lymphatic, and occur between 10 to 90 days, 3 to 4 weeks. Your dissemination resulting in the hallmark syphilitic region, tuvuga ngo ni chanco, which is painless and is more form or more solid than the one that we have seen with with Haemophilus decree, which is more soft. Chanco change from hard to ulcerative with a profuse shedding of spirochetes. So it's change to ulcerative, bifite nuze nyinshi za spirochete, and swelling capillary walls and regional lymph nodes. And the lesion can heal spontaneously within 2 months, leading to 4 sense of relief. So munagira ngo arakize, so it really give a false sense. Munagira ngo arakize, or it's really going to be worse. Secondary disease, 2 to 10 weeks after primary lesion. It's more widely disseminated. Murareba utuntu twose, utu region twose twuzuyemo za spirochetes. Secondary region of the skin and the mucous membrane are highly contagious. So they are very contagious, full of spirochetes. And we have generalized immunological response. So in this case, we have even the cell-mediated immunity, and this can also go spontaneously. Shobora kwirangiza kubera ko immune system irazica izi spirochete cells zose ziri infected. Then come a latent stage syphilis, which is without symptom. It can be first 4 years, which is early latent, subsequent periods late latent. About 40% of latent patients progress to late tertiary syphilitic disease. So, nokuvuga ngo sometimes after the latent stage, some can progress to tertiary, some others can cure spontaneously, cyangwa zikaguma ziri latent for longer, cyangwa soon. Tertiary syphilis characterized by localized granulomatous dermal lesion, izo twita gumas, in which few organisms are present. Ikiba gikora ubu ni immunological reaction. So the immune response contain the spirochete ntago ziba zikidividinga, ziba ziri contained, ariko immune reaction is also harmful to our cell. And late neurosyphilis develop in 1 to 6 untreated cases, usually more than 5 years after initial infection. So easy cases zishobora kuza, ariko sometimes they are rare, they are not such common, and they affect central nervous system, spinal cord involvement. Patients will have wasting, seizure, dementia. Sometimes cardiovascular involvement appear 10 to 40 years after initial infection, with resulting myocardial insufficiency and death if happen. So it comes late, ikaba yatera insuffisance cardiaque, iganisha kugupfa rapidly. So we have the flow like we just discussed. We have that up to 8% may be develop central nervous system syphilis, which is late stage or tertiary syphilis, 10% cardiovascular complication, and 15% late benign or without symptom. Sometimes spontaneous cure is possible at any stage. Ishobora kuba cured spontaneously by the immune system. So these have been well studied. Nkuko twabivugaga ubushize, hari study yabayeho yabaye more ethically controversial where people with syphilis were more intentionally exposed to infection, and they follow them up without treatment. Nubwo treatment yabonetse, penicillin yaje kugaragara ko effective, ntabwo bigeze babavura. Bashatse ko bakomeza kubakurikirana bareba uko syphilis igenda, badescribinga stages zayo batavuye abo barwayi, which is really unethical study. Yatumye habaho ethical as rigorous ethical committees as it is now mu by'ubushakashatsi. So congenital syphilis is also something that happen generally through transplacental infection when the mother is infected, and septicemia in developing fetus and widespread dissemination. Kenshi na kenshi it results in neonate mortality and abortion. Sibyo, kenshi umwana ntavuka, it lead to death, and sometimes niyavutse avukana mental and physical disabilities. Mental and physical disabilities resulting from the scar from the active disease and progression of the active disease state. So having such condition, umurwayi cyangwa umwana, they can't develop. So we have different methods of diagnosis, mostly dominated by dark field is not everywhere. You cannot use it, and culture not available, not done. So we rely on serology. How to fit a non-treponema test, dukora, rapid plasma reagent, cyangwa venereal disease research laboratory. Izi zombi zigakora screening, then we confirm with a treponema test, nk'iyi first and treponema antibody absorption test assay. So these two can confirm the cases in here. Sometimes you have false positive serological tests for syphilis, aho non-treponema test zishobora kuba positive umuntu afite malaria cyangwa leprosy, cross reactivity, cyangwa treponema test igihe umuntu afite izi condition. So it's again important for clinician to really understand the symptom of syphilis and try to deduce the clinical diagnosis based on also the reactive tests. Prevention and treatment of syphilis, penicillin is very effective, strong. WHO monitor treatment recommendation 7 to 10 days, continuous for early stage, at least 21 days continuously beyond the early stage. No kuvuga 21 days iyo umuntu azifashe, it really get cured. So prevention, gukoresha condom, kutabikora, kwifata, or you can also do prophylactic treatment of contact through epidemiological tracing. Niba mushoboye kuidentifya ka imwe ya syphilis, mukaba mwavura network y'uwo muntu yose kugira ngo mu contracting disease na dissemination yayo. The second genera is Borrelia, where we have Borrelia recurrentis, which is the main or the most important, caused by body loss, epidemic relapsing fever, which is louse-borne relapsing fever, with human as the only reservoir, while the other Borrelia have other reservoir. Borrelia recurrentis reservoir yayo ni muntu gusa. So it is transmitted by body loss, and this is Pediculus humanus. We have other Borrelia species. The relapsing fever is endemic. So we have other reservoir. In soft shell tick, they are the one transmitting this Borrelia species. We have Borrelia burgdorferi, the causative agent of Lyme disease, and Lyme disease is more of transmitted by hard tick, hard shell tick. So relapsing fever, which is caused by Borrelia recurrentis and other Borrelia species, is associated with poverty, crowding, and warfare, arthropod vector, nk'uko twabibonye. Rero poverty cyangwa crowding ihari, inda ziraza, then this kind of disease is then becoming more epidemic. Kuko umwe iriye ijya ku wundi, especially when the person develop fever. So the louse we have tendency to get out of the case with fever or the body with fever to infect another person who seems to be normal. So the infection does not come from the mouth of louse, rather injured or killed, scratched louse, like during scratching, and the Borrelia will just enter the area where the louse is perforing the skin. And this is what we say, once the host develop fever, they will seek for a normal temperature host. Zimuvaho nyine zumva ko ubungubu ibye byarangiye zikajya gushaka undi. So by staining with Jamusa, you can be able to see the spirochetes in blood, and phase contrast as well, urayibona easily. Endemic relapsing fever, a tick-borne borreliosis, is also sporadic, transmitted by soft body tick, especially from small mammal reservoir. And pathogen, so they transmit. Infected ticks can transmit it is like hereditary. Once it gets infected, it can spread the reservoir throughout the progenitor, and the progenitor can keep infecting people. So there is a acute infection with 2 to 4 days incubation, and followed by recurring febrile episode. Recurring kubera ko once the immune system is developed, it will really kill most of spirochetes. Some will undergo antigenic variation, leading to another cycle of multiplication, increasing fever, and of course evading immunity. Your immune system will established kuri new strain or new the antigen variant. It will again suppress the growth of pathogen, but eventually it will keep changing the antigen. So that is why, or the origin of recurring febrile episodes ari nabyo bituma tuyita relapsing fever. So it keep increasing, reduced, increase, depending on when the antigenic variation takes place. So Borrelia burgdorferi is the causative agent of Lesion. It starts with initially unique skin lesion, erythema chronicum migrans with general malaise. You don't have too much problem. Subsequent stage is rare. It can be seen up to 15% with neurological, even cardiac involvement. Third stage involve migrating episode of non-destructive but painful arthritis. So it can even cause major condition like arthritis. So there is a clinical diagnosis is more important. Our clinical case definition have to be well mastered, and well good clinician we already suspect the disease, and at least one of the following criteria should be positive to diagnose the disease. Either grow the Borrelia in culture cyangwa IgM IgG test. You can also do the antibody titer, or the level of antibody. Another is the agent or the causative agent of leptospirosis, which is obligate aerobe. If it a hooked end, like a question mark, niyo mpamvu bayita interrogans, Leptospira interrogans. So they have mild virus-like symptom, anicteric leptospirosis, tukagirana iyo iri systemic with aseptic meningitis, tukagirana ecteric leptospirosis, which is a very overwhelming disease, also called Wells disease. It causes vascular collapse, thrombocytopenia, hemorrhage, and hepatic and renal dysfunction. So it's really a very dangerous disease, but rare. It's not such very common. So you can see the pathogen in the specimen. So it's also called Wells disease in leptospirosis, and direct invasion and replication in tissue, the pathogen leptospirosis just replicate. infection is common. Nayo nk'uko twabibonye igira ziriya cycles za disease, and igira leptospiremia, igira meningitis, igira kidney involvement causing a very serious disease, renal failure, cardiac myocarditis, jaundice, and hemorrhage. So it's a very serious disease, although it's more rare. It's a rare disease, and it's mainly a zoonotic disease. So it's kind of not transmitted between person to person, rather than having the infection from wild and domestic animals. Niho abantu bakura infection. Reservoir kenshi ni rodents, like rats, dogs, farm animal, and wild animal. And it's transmitted through a break in the skin or intact mucous membrane. So once it's access the intact mucous membrane cyangwa skin with abrasion, it just gets in. And nk'uko twabibonye, it is shed in urine. So the urine from an animal with a leptospiria, we directly transmit this either muri soil cyangwa water cyangwa food. It is more occupational disease of animal handling agents. So it's more in people handling the animals. So it's quite easy to diagnose in microscopy. Cannot see the organism, of course. Dark field is a bit insensitive. Culture of blood, put it during first 10 days. Cerebrospinal fluid as well, urine can be cultured. We can do nucleic acid probe, PCR or direct hybridization. We can do serology, so we can detect the antigen or antibody of the pathogen. So any question? Any question? If no question, I think it's good time to break a bit, and we will continue on Monday afternoon. If time allow, we continue Monday afternoon, and what is remaining is more to show some aspect of practical sessions. And Virako, some of practicals, you are conducting practicals with Nicola, and I will have an overview of possible practicals nk'uko muzifite kuri slide tuzinyuremo. If some questions are pending, I can still explain, but we will not take too much time on slides, rather than allowing you to access the lab and have exposure on the lab. So any question? How was the cut? The cut was easy.
Very difficult.
Kati yaratanze kabisa.
Was it easy? Was difficult?
It was so difficult.
It was so
Yaratunaniye.
what?
Yaratunaniye kabisa.
Yarabananiye?
Yaratunaniye.
Nibyo? Na virology?
Biology ubwo niyo yatunaniye cyane.
Okay, so maybe we do better in exam. Abantu batakoze cat, nako assignment, I really want them to come back with hari topic nifuza ko nabaha on Monday, then they can work on it and present on next Friday, bakagira presentation ntoya kugira ngo tubone uko bazabona amanota nabo. Otherwise, it will be difficult. Okay, bye. Have a nice weekend.
Thank you.
Likewise. Nice weekend too.